Hi everyone, welcome back to the new video. In this video, we are going to focus on a topic called as IgG antibodies. So in this video, we are going to discuss regarding the structure, function and clinical significance of the IgG antibodies. Immunoglobulin G, that is IgG, is the most abundant antibody in the blood and extracellular fluid. So it plays a very crucial role in immune defense mechanism. So it is, of course, it is a Y-shaped protein produced by the plasma cells, activated B cells, and it actively responds to the infections and vaccines or other foreign substances, that is antigens. Now let us see the basic structure, or you can say the structure of IgG antibody. Actually it is a monomer and it contains two heavy chains and two light chains. And here the new terminology, that is KDEA. It has 50 KDA and it has 25 KDA. So in this diagram we can see there are heavy chains and light chains. So these are the light chains. So these are the light chains. So here we can see the light chain. So here also we can see the light chains. Okay, and this portion, they are the heavy chains. Okay, then these light chain and heavy chain both are linked together by a bonds called as disulfide bond. Here we can see the bonds. Both are heavy and light chains, both are linked together by bonds called as disulfide bond. Here also we can see the bonds. They are the disulfide bond. Now what is the meaning of this one 50 kDa and 25 kDa? So this one having 50 kDa, that is heavy chain having 50 kDa, and light chain they are having 25 kDa. So what is this KDA means? So KDA is nothing but a dissociation constant. Here dissociation constant means it will determine the affinity of the antibody. To bind to the antigen. So here antigen means any foreign material which is going to trigger the antibody production is called as antigen. So this KDA, that is dissociation constant, that is 50 KDA and 25 KDA, it will determine the dissociation constant, means how much it is having affinity towards the antigen. More the dissociation constant, having the lesser affinity towards the antigen binding. So here dissociation means here separation. So if the antibody having the more dissociation value, that is KDA value, it is having very less affinity towards the antigen. And if it is having lesser KDA, that is the dissociation value, it is having more affinity towards the antigen, and it is having strong antigen binding capacity. So now we are moving towards the other parts. That is, another thing is there. Here we can see that is hinge region is there. So hinge region means this portion. It will provide the flexibility. See here we can see the hinge region. So these are the bonds, disulfide bonds. So this is the these are the disulfide bonds. So here we can see the disulfide bonds. Sorry, so this is the disulfide bond. Okay, and hinge region means so this portion is called as hinge region. So this is the hinge region. So hinge region, it will provide the structural flexibility. It will provide the structural flexibility. Structural flexibility. Structural flexibility for the antibody to move. So it will provide the structural flexibility for the antibody in order to move. So this hinge region is mainly responsible for the flexibility of the antibody. And the remaining thing in this structure is here we can see the FC region and Fab region. FC region and Fab region. Fab region and FC region. So here we can see here is the Fab region. That is Fab region. Here we can see the Fab region. That is FAB, Fab region. And here we can see the FC region. So this is the Fab region and this is the FC region. That is FC region. So a Fab region means that is antigen binding site. That is here is the place where the antigen is going to bound or attach to the antibody. That is Fab region. And constant FC region is the effector region. Means it will determine the mechanism of the antibody, how the antibody is going to kill the antigen, or what is the mechanism, what is the procedure which is going to adapt by the antibody in order to destroy the antigen. So effector region, it will stimulate or trigger the complement system. Complement system means, as I told in the earlier videos, a complement system means it is a group of proteins which is going to play a vital role in destruction of antigen. It is a group of protein which is going to play a vital role in destroying the antigen. That is, it is a combined work of the WBCs to destroy the antigen. That is called as complement system, complement activation. So these are the some different parts of the IgG antibody. Means that is 2 heavy chains and 2 light chains and 50 kDa and 25 kDa. That is dissociation constant. Dissociation means separation. If the more the value of dissociation constant means it is having lesser affinity towards the antigen. If it is a lesser kDa value means it is having more affinity towards the antigen, and it will bind strongly to the antigen. And and these chains are linked together by disulfide bonds, that is SS bonds, and having it is and another two region, that is Fab region and FC region. Fab region is the region where the antigen is going to bind. FC region is the thing which will decide the function or mechanism of action of the antibody. That is called as FC region. Then hinge region, as I told you, hinge region is the one which is going to provide the structural flexibility to the antigen. Okay. Here is the subclasses of IgG antibodies. Here is the 3 columns are there. In the first column, the name of the antibody, subclass, and in the second column, the percentage which is present in the serum, and in the third column, the main function or the key feature. So the first one is Ig1. So it is about 60 to 70% is present in the serum, and it is most abundant, and it is present in excessive concentration, and it has a strong complement activation property. The second one is Ig2. and it has about 20 to 30% in the serum, and effective against polysaccharides and antigens. That is example bacteria. Sorry, polysaccharide antigens. Example bacteria. Then Ig3, it is about 5 to 10%, and it has a strongest complement activation system. That is, and it is having a longest hinge region. Then Ig4, it is about 1 to 3%, and it is having weaker effector functions involved in allergies and immune regulation. Functions of IgG antibodies: neutralization. IgG antibodies binds to virus, toxins and bacteria, and blocking their ability to infect the cells and causing harm to them. The second function is how the antibody is going to destroy the pathogen, means by a simple process called as opsonization. So what happens in opsonization? So here the pathogen or antigen or microorganism is get tagged by the antibodies, then it is easily recognizable from the macrophages, and the macrophages get they come and engulf the pathogen or antigen or disease causing microorganism. Complement activation, especially Ig1 and Ig3, activate the classical complement pathway, which is going to cause the lysis of the pathogen or destruction of the pathogens. So now what is complement activation? Complement activation helps the antibodies by creating a team of proteins which are going to work together to fight against the bacteria or pathogen or viruses. How it is going to work? It works in a different ways. In among them, the first one is it is going to spot the invader, means the antibodies will spot the pathogen, or it is going to code the pathogen, or it is going to make the pathogen more recognizable, easily recognizable by the immune system. And the immune system, that is WBCs, they are going to detect the harmful germs, and they attack on the pathogen and kill them. Another way is this complement system activates a group of proteins, a special group of proteins, and these are very powerful proteins, and they cause punches holes in the germs and destroy them. Then after destruction, the immune system comes and make it clear from the body. These pathogens are removed from the body, and the damaged invaders, that is damaged pathogens, get cleared from the body. So this is about the complement activation, and this complement activation actually it acts like a alarm that calls for the antibodies, and it will help the antibodies to fight against various types of infections more effectively.